Thank you so much for attending my talk. I'm really excited to have been invited and to share with you all what we've been up to with our next generation scientific R&D platform called Luma. Our specific application here today that I'm going to speak about is revolutionizing a therapeutic antibody engineering space, the whole R&D space with Geneious Luma. That this really is a path to next-gen multi-specific antibodies. So our agenda today just breaks down into 5 main parts: an introduction, challenges, the way that we see them, the recipe for innovation that we've developed, the pattern that we've identified, and I'm going to take some time to do a deeper dive into Geneious Luma. So by way of introduction, my background, I've been within the biotech space for quite some time. I've had a fantastic number of different positions across these organizations on the right, but this is really what has brought me to Dotmatics. So my background is molecular biochem, bioinformatics, infectious disease, drug target selection, and I've really been able to take all of this to help inform the way that Luma has developed and what it's going to provide for our biotech and pharma customers. So the challenges as we see them, there are a number of challenges, but first and foremost, the modes of therapy have multiplied versus 30 years ago. Biology is really complex, and there have always been issues about data flow and access. And when you combine these three together, you really have kind of a perfect storm of difficulty as you're looking to develop new therapeutics. So the figure on the right is just an example of the complex biology of COVID. So this is just a phylogenetic tree of how different COVID strains evolved over time. And it just really shows you that it's hard. So we also have other challenges, data visualization, data flow, data tracking. And in terms of visualization, it's not just visualizing the data, but underneath that, it's really managing and filtering large data sets. So typically in this space, you need to have some type of expertise with databases. You need to have bioinformatics expertise. There are learning curves associated with learning software like R or Python. And you have a lot of different data types that you need to work with. For flow of data, it's well known that data are siloed. And this really impedes the collaboration within and across sites. And it leads to repetition in experiments because prior results were misplaced or disconnected. For workflows, for tracking, really, it's been difficult to track who did what and when did it happen and what the results were. And so oftentimes you get a lot of duplication within the process, which is wasted time and money. So in terms of therapeutics, there are challenges. So the modes of therapy have multiplied. They have N levels of complexity that are only magnified by our ever-increasing understanding of human biology. And so that's really why we developed Luma. Luma is the first platform capable of handling not only the multiplicity, but the complexity of multimodal therapeutics. And closely coupled to Luma is Luma Lab Connect, which is an integral part of moving data from your instruments into a highly performant data store so that you can bring together data from all of your applications, all of your instruments, and your files. Now, today's talk is going to focus on multispecific antibodies. So the challenges associated with those is combinatorial. R&D has really been limited to a small fraction of multispecific antibody molecules. There's not been enough time or money to explore the whole combinatorial space, and really software tools haven't kept up. So because of cost, scientists are pushed to not test for diversity. They only test for a small fraction. And so this is due to the major technical limitations for testing 10,000 different multispecific antibodies at a time. And so when we're talking multispecific antibodies, just taking one step closer, we're going to talk about like a tri-specific antibody. Okay, so the cartoon on the right, the left-hand arm of the antibody binds two different targets, and then the arm on the right binds one target. So you have three different targets that you're going to join together to a knob and hole region at the bottom. Okay, so if you're focused on CDR sequences, which everybody is, you're going to focus on CDR 1, 2, and 3. And say you've got a CD3 sequence or CDR3 region, and you've got 55 candidate sequence that you want to test for that CD3 region. Same with CD28. Maybe you've got 50 candidate sequences, and CD30, 38, you've got 20 candidate sequences. Okay, so you have that range of sequences, and then you have maybe 5 sequences for the knob and hole region at the bottom. Well, if you wanted to join all of the combinations together, you've got over 250,000 molecules that or combinations that you could test for. But this has never really happened because it's just too many. And so, a clear example from a very well-known publication came out. I've just blown up a small fraction of the way some of these molecules are put together, but the combinatorial nature of the multispecifics is vast. Okay, and so hopefully, this resonates with everyone. And then with this, we have to talk about chronic disease. So if we're going to treat something like cancer or metabolic disease or a whole host of things, there are a number of different causations which can either work independently or in concert with 2 or 1 other causation elements. Okay, and then there's your immune system, which is incredibly complex. And then there's time. So perhaps you were infected in utero, and your chronic disease doesn't manifest itself until you're 55. Well, tracking that disease path over that amount of time is incredibly difficult. So all this relates to our biologics drug development over time, where we've got a ton of candidates in the basic research space, and the attrition rate between basic research to FDA review is just simply massive. And actually, it's unacceptable, right? I think we can do better, and that's what we're doing with Luma, right? So we're broadening the funnel for basic research so we get a larger collection of molecules to test, and hopefully more preclinical candidates and a greater amount of FDA review at the end of all this. So next, I want to talk about the recipe for innovation that we have put together. Okay, so in the past, this is how we've thought. We thought in a very fractured way. We manage ELNs, electronic lab notebooks. We manage LIMS systems and inventory systems, instrument integrations, registries, and on and on. And it's a very siloed and fractured way of thinking about things. And this is the impetus for Luma. Right, this was the best we had for many decades ago, but we can do better, and we have done better with Dotmatics Luma. So the pattern that we've identified as we've talked to customers, as we've been in the laboratories, as we've talked about major pain points of R&D teams, the pattern that we found is really about composability. Right, so scientific research, scientific workflows can be composable, and scientists compose the units of research differently based on their use cases. And so we developed just a very, interesting and incredibly important, but smaller slice of what we mean by this. So if you're doing antibody discovery, whether you're doing phage display or hybridoma or using transgenic mice, or you're working directly with B cells, or if you're working with oligotherapeutics like with siRNA, these research units have a lot of commonalities, even though they're distinct. And then we're talking about instrument data, where you want to look at ELISA assay results or LCMS data or flow cytometry data. All of those things the instrument data flow into Luma Lab Connect. And over here in the cartoon on the right, we've got Geneious, and we've got fully modeled data. So we've got a way that the data all relate together. Okay, so the Luma units naturally fit together to solve a diversity of workflows, and all of these again are derived from real in the wild customer use cases. So other scenarios like vaccines and CRISPR therapeutics and cell therapy workflows, these are also supported too. So to talk a little bit about Luma, Luma is a multimodal scientific intelligence platform, and we've got things broken out into 4 quadrants. So the top left, we have a blend of scientific and IT disciplines. So you're working with chemistry, small molecules, or biology. Maybe you're working in material science. All of these have an element of data science. All of these have an element of computer science. And all of these have an element of administration from the IT teams. And at the top right, we've got a number of different modalities. So we've got small molecules, antibody drug conjugates, vaccines, and so on. Bottom left, we have a lot of different scientific techniques. So we're working with assay data or sequence data, protein characterization data or flow cytometry data. And then over on the right, we have different data modalities. So we have structured and unstructured data, ABML, SBML files, multi-omic and clinical data. And really all of these 4 blend together into Luma. And we've really, I think, elegantly just kind of thought about this as you're making things, you're testing things, and then based on those test results, you're making critical decisions. So Dotmatics Luma really is about accelerating insights, really empowering the users for the best decision-making tools you can have, as well as to improve the outcomes. So over here on the left, we have instruments, and instrument data stream into Luma Lab Connect, where the data get parsed and stored. And those data are accessible by Luma, as well as all of these scientific tools that are in orbit around Luma. So tools like Geneious and SnapGene and Prism and Omic and Protein Metrics, right? All of these best-in-breed tools are able to interact with their data type when they need to. So we're really empowering our users to have access to cross-modality and conjugates R&D, to have contextualized decision-making at every level of their process, to be able to visualize and analyze really well-correlated and complex data. Oftentimes we pick up patterns with our eyes way quicker than we do with our Excel spreadsheets. And we really are providing an enrichment of existing system via open APIs. So we strongly believe in having an open system. Okay, so you can interact with Luma using APIs with standard methods like JDBC or GraphQL. We have a whole host of RESTful services. Like we have a whole host of ways you can interact with Luma. So, to dig in just a little bit deeper about instrument and data integration in Luma Lab Connect, this is really what it looks like in the wild. So the data on the left stream into Luma Lab Connect. At any given moment, you can see how many instruments are online. You can see how much has been uploaded. You can see the size of the files and the last sync and what platform. And so really, this is about your data and your data coming together at the right time, in the right context, with the right structure and the right scale. So this is critical data management and processing, right? So Luma Lab Connect is the link to unlocking the value of your data, right? So you have Luma Lab Connect, parse data, that's semi-structured data that you can model and you can have structured, and then you can correlate those data. And these correlated data can feed into tailored user experiences, your own internal systems if you want, our adaptive workflows, which I'll talk a little bit more about. So really unlocking the value of your data. So Luma Lab Connect, it's super quick and it's very simple to deploy. You can deploy to hundreds of instruments quickly and, think days, not months. You can remotely manage your updates from the cloud. You can centralize data streaming from machines. You can land those data in flexible data models, which you can version. So built-in data modeling tools really move you past those antiquated file-based approaches. So you can quickly and efficiently correlate data from multiple streams, and you can build a complete landscape, a tapestry of your data, right? And all this so that you can accelerate decision making. You can make better decisions about therapeutic candidates more quickly. And this includes user-defined dashboards and data experiences. So Luma is about using the best of breed tools that you love, that you know, that you've standardized on, or you can use your software. You can include your favorite analysis software or that instrument that hasn't been invented yet. It'll come out next year. As long as it has an API, we're able to move the data from that instrument into Luma. So Dotmatics enables this lab in a loop concept where you have connected, harmonized, structured, fair data that feeds models. Okay, so you have experiments that produce your data that move into connected, harmonized, structured format. And then in turn, you're able to utilize AI and ML models, and insights really guide the next experiments that you glean from the experiments. And so those insights you feed back into the loop. Okay, so you have a portfolio of applications, you have an integration and organization layer with scientific expertise and data management capability, all very critical. And then you have algorithmic approaches and the opportunity to recognize patterns and leverage cloud compute, and really to enable AI and ML. Until you've gone through digital transformation, AI and ML really is not available to you. Okay, and so we've directed this at an AI native multimodal drug discovery platform, regardless of the modality you're working on. Okay. So I'm going to take one step deeper for application and talk about a B-cell antibody workflow in Luma. And the only things, the text is really small, but the two things I want you to take away are this. We have an outer perimeter of tasks, and the input of one task produces an output for the next task. So what I'm showing you here is we've got, you've done your B-cell sequencing, then you need to annotate your sequences with variants and liabilities, and then you need to filter those sequences, do some analyses on those, and then once you pick the interesting ones, you send them to your cloning group. And your cloning group needs to make constructs using plates. Those plates generate data. Those data move into Luma. From the cloning group, you send those, or you perform QC sequence validation. Then you send those to your protein expression purification group. And then step number 5, in the far right in the middle, you run those sequences or those proteins through some battery of screening assays just to, further filter down into the highly likely sequences. And then you associate those assay data with the sequences. And then you go through another round of cloning and optimization. At the bottom, step number 8, another round of sequence validation, expression purification, and then a deeper level of screening assays. Okay, so you have the outer perimeter, and then you have this inner perimeter where off of these tasks, you have various plates that are made. Those plates generate data. Those data need to be in a centralized store, which is Luma. Okay, well, I'm talking to you about Geneious Luma today. So in Geneious Luma, you can import your FASTQ files or Sanger data or 10x data. Regardless, it doesn't matter. But you're able to annotate those data. You're able to cluster those data based on regions. You're able to filter down into the data. And then you're able to send those data to Luma. So the act of choosing sequences and then clicking on register in Luma, that registers those sequences behind the scenes in Luma. And those sequences land in Luma and are able to be picked up by your cloning group. Okay, so Geneious Luma also allows you a variety of cloning methods to do recombinant cloning for those antibody sequences. So regardless if you're doing, generating monocistronic plasmids or bicistronic plasmids, you can do both with a host of tools, in silico tools, and you can do them in bulk, right? So you've got 1,000 constructs you want to produce, so you've got a monocistronic situation, which I'm showing you right here. And, once you've generated your constructs in bulk, you can send those constructs and register them in Luma, right? So they land in Luma so that adaptive workflows can now pick up those constructs, and you can begin building or specifying your experiment with those constructs in 96-well plates. So adaptive workflows looks like this. Okay, so once you've got your constructs registered, then adaptive workflows is really the workflow tool that you set up your containers, your materials, what the plates are going to look like, what's going to be in each of those plates. You have a plate view, you've got a table view for what you're expecting versus what was actually made. And so adaptive workflows really is the glue that binds all of the scientific R&D processes. So you're setting up your scientific tasks, and each of those tasks you can configure, and this is what it looks like. Okay, so let's step into the weeds. So we're talking about multi-specific antibody design. So we are delivering a tool that allows you to design and register your multi-specific antibodies. You're able to label the data, and you're able to implement AI engineering, in silico assessments of those molecule formats. And what I'm showing you here on the right is the design pad that you can use to design different multi-specific antibody formats. Okay, so in the middle, you see your classic IgG antibody structure, and at the bottom, you see two scFv sequences that are joined. Okay. So this is really driven or empowered by two different really important groups. I just wanted to recognize the Martin group out of England that developed the antibody markup language. But then there's an Amgen group by Biswas or Russell et al. That developed the Veritas antibody language, which is a little bit more human readable than ABML. And the antibody markup language is really important to describe the multi-specific format that you see here on the left. So you've got a Fab building block joined to an FC building block joined to scFv building blocks, and you can describe those using ABML notation, which I'm showing you here on the right. So you have single entities that in the past we've been forced to work on. Well, with Geneious Luma, you can go from single entities to panels of entities across the sequence space. So just a quick example here. Over here on the left, we've got two different type of building blocks, 10 types or 10 sequences for each type. And then moving over to the right, you could have format number 1, where you've got scFv connected to the Fab. Format number 2, you could have that scFv joined to your FC region. And this could generate up to 1,000 molecules, okay, just as an example. And so really we move through kind of the make, test, decide cycle with these multi-specific antibodies like this. You design your formats in silico, you assess them, you get the results of those, you score them in silico. You have a number that pass, a number that need to be engineered, and then a number that fail. And for those that need to be engineered, we call that the rescue path. So you can send those back to get them engineered to improve their characteristics, send them back into the assessment, and hopefully recover additional molecules. So we're testing for molecular fitness before they even go into the lab. And so this really expands initial screening. And so over here on the right, this is the multi-specific format that we designed, top left, that moves into molecular dynamics machine learning in silico assessment. Those molecules are scored with this heat map down at the bottom left, or bottom right, I'm sorry. And then any that pass, we move on. Any that could use some engineering, we can feed those into different models. And then from there, we can assess them again. Okay, so we're really hunting for or wanting to aim for superior biological properties of these molecules before we ever go to make them. And we're looking for safety and efficacy and desired pharmacokinetic properties. But also, downstream, we want to optimize the developability profile. So we want to have a, manufacturability assessment stability and regulatory and even for drug delivery. Okay, so again, the complexity comes to the surface, right? And so if you can do the bulk of this in silico, you can save a lot of time, a lot of money, and you can increase the number of sequences that you're able to test. So here we're showing the multi-specific AB panel generation, where you've got a bunch of different type of formats across the top, and then the building blocks down at the bottom. You have different building blocks with sequences associated to them. So you've got a Fab A region, you've got a Fab B region, and at the very bottom you have a hetero FC region. And so now we're working at the panel level. We're not just working at 1Zs, 2Zs. We're working at panel Zs, right? So if you've got 5 panels, 10 panels, 30 panels, you can really generate and account for all of the combinations of molecules in one place. And so really, Luma unlocks innovation. You have linked multimodal data combined with a maximally performant database that is precisely structured. So when you're working with all of those multi-specific antibody formats and all of the underlying sequences that are associating with them, and you're exploring all of the combination space, you really need to have these elements on the left in place. And this really sets you up for AI and ML applications. And so Luma is the AI-enabled multimodal platform. That's what it is. So you go from isolated data points from applications and instruments. You then move to connected data from applications in a workflow, right? So you've got a portfolio of applications. You've got an integration and orchestration layer, deep scientific expertise, and then a very, very effective data management capability. And that enables you to have computationally enabled data. So that's where you can take algorithmic approaches, you can do pattern recognition, you can leverage cloud compute, really enable those AI and ML applications, and you can point all of those to the therapeutic discovery space. But it's not only about antibodies and biologics, right? This is where we've started, but this caters to a whole host of other therapeutics. It also caters to drug discovery support. So whether it's protein production or library development or pharmaceutical formulations, you can leverage Luma for drug discovery support. But also here on the right, there are a whole host of other applications to be explored: chemicals, materials, agritech, public health surveillance, battery technology. So Luma is a very broad platform that can be tailored to suit your specific workflows. Okay, your entities that you're interested in for your applications. And so I want to take a moment to acknowledge my team: Kaleem Saliba, Michael Schwartz, Scott Stunkel, Fernando Garces, and Richard Moir. Without all of my team members, as well as a whole host of others underlying, Luma wouldn't be possible. So I really wanted to acknowledge these people. If you'd like more information, you can reach us at luma.dotmatics.com. Really, we are very excited to deliver this platform into this space. We're eager to talk to you, so please email us if you have any questions. We would love to talk about your use case and what you're working on. And I just want to thank you very much for your attention today.